Okay. Yeah.
What did you choose DUS1 over the other ones?
Because they weren't inhibitory. And some of them basically induced DUSP and they were going down, so that really, I think for my purposes, and this is just kind of a personal, I've been looking at this data so long, is that when you see something like that, you see a bunch of red and then you see it all funneling into something green. It's usually an inhibitory molecule that's like blocking everything. Some cop showed up and went, no, we'll not have any of that, and took it away. That's kind of how you figure that stuff out. And as you get into your own projects, you're going to find things like this. Bring me over and say, hey, Edwards, look at this. Does this make sense? And I'll go, oh, yeah, this works. Or how about that one? What you'll find is it's not only like, am I looking for inhibitors, especially when I see that bottleneck, but what I'm looking for are things that are connected to lots of other things. And that kind of like when we get into these networks, what you'll see is you'll see one like thing that changes and it interacts with a lot of different things on your gene list. It's like the bomb, it's like your godfather interacting with all its soldiers. Why would I take a soldier out when if I get the godfather, I get rid of all the gangsters, right? That's kind of what we do with this gene expression stuff. Okay, canonical pathways, IL-6 signaling. Honestly, you can look through here. This makes perfect sense. Sertoli cell junction signaling. Anybody know what a Sertoli cell is? Down below. Testicles. Yeah. It's basically how you form sperm. And in order to form sperm, you have to compartmentalize the testes. And so you need a lot of cell adhesion. So basically when I see this, it never fails. Like I'll bring this list up and somebody will go, well, I'm not working on testes. I'm like, well, that's okay. What this is showing us is that you have a lot of cell adhesion, or at least cell adhesion molecules that are either going up or down in your system. We can click on this and you'll see it kind of. You see what the network looks like. Yeah, everything's connected. Knowing nothing about this system and knowing that, green is down and red is up, what do you think is happening to the connections here in these cell types, in the pluripotent stem cells? Look at all the green on the edge. These are connections here. They're all going down. What this is telling me, it's giving me validation on our biological function thing is, yes, cell adhesion is probably reduced in these pluripotent stem cells. And again, we can modulate this any way we want. We can stick a molecule in here, say that, go to your build function again. We can select, this time go add molecule relationship. In this instance, we can add anything we want. We're going to tell it what it is, so just pick biological drug, that's fine. Okay, so you should have a dot here. Once you click on your pathway, like a blue oval will show up. Double click on that. All right, well, P53 is supposed to play an important role in, stem cells, right? It's all about, higher levels of P53, more differentiation maybe. Let's put in TP53. That's its official gene symbol. Now hit search. All right, you're going to see a bunch of stuff come up. Why don't you go to about, I don't know, it's like almost 2/3 of the way up, maybe. There it is. Hit P53. It's not the one with a cross on it or whatever the heck that is. It's the one right below that. Hit select. Now hit OK. We have now inserted P53 into our pathway. Now if I want to determine if anything in this pathway is affected by P53, here's what I do. Is I go back to build, and then I go to connect. Usually I'll leave all the defaults. I want all direct, indirect. I could, though, basically specify, maybe I only want to see relationships at P53 and say endoderm, right? I could do that. I'm going to leave everything the way it is. I'm just going to hit apply. I didn't do this in practice, so let's see what happens. Yeah, it's a little bit connected. What do you think? Isn't that cool? No gasps, no. Everybody's like, we're tired, man. We just had like diff EQ like yesterday, man. I think this is cool. Not only that, but we could color this. We could figure out what kind of relationships. Again, this is a good way to determine, hey, I've got a gene of interest. Does it have any relationship to whatever I'm looking at? Okay, click out of that. I spent too much time on pathways. For somebody who hates pathways, I spent way too much time on that. Okay. Let's go to networks.